Okay. Everybody, we're going to be jumping into Biochem 1, studying for our final exam. Most important thing, if you don't have snacks, get yourself some snacks. Your brain cannot operate if it is not properly fed. So, with this, what we're going to do, we're going to try and fly through the first chunk of material. What was on kind of exam 1 and exam 2. Hopefully a lot of this at this point is very repetitive and very familiar to all of you. So hopefully we shouldn't have to spend a lot of time on it at all. As we get into more of the newer stuff, we'll slow down a little bit, hit some of those key points. And then again, as always, if you have any questions or comments or concerns, throw them in the comments down below. Let me know so that I can go back, clarify anything if you have any questions or anything specific is a little bit confusing. Also, in the description below, you'll find a link. Reminder that for the entire program, I have made available all of my notes. These, again, I'm a very meticulous note taker, as I'm sure you can tell at this point. And I have continued this trend all the way through the entire program, and kind of this making those available for a very, very cheap price for as many students as I possibly can. It's mostly for right now to provide for my family during this time at school when I can't or I don't want to do an outside job to take away from my studies and support my wife and my son. So if you're interested, links down below, and that will always be there. So yeah, feel free to check it out. Okay, so starting with the very first lecture, the chemistry review, the ions that are formed, we have positive and negative ions. Hopefully this is all super review. Cations are positively charged, so they've lost an electron or they've gained a hydrogen atom. Stuff like potassium, sodium, calcium. Calcium is a 2+. Potassium and sodium are 1+. Keep that in mind. And the anions are negative. The most common one we're going to see is oxygen. That's our big one that we're going to see in all of these reactions, and those gain electrons. So different chemical bonding. Ionic bonds are a big one that we're going to see a lot of. In this one, a cation and an anion, so positive and negatively charged, come together, form a bond, and those are fairly strong bonds as well. So covalent bonding is when the atoms share valence electrons. So around every single atom, you're going to have valence electron shells. And in those valence shells, you have electrons, and they want to have what's called a full shell. The very first shell has 2, the next one has 8. I believe after that it's either there's another one that's 8 or it's 16, and so on and so forth. It continues to go up. So they want a full valence electron shell. Their last shell they want to be full. So either they're going to shed electrons or they're going to gain electrons. And that's kind of what covalent bonding does, is that it pairs up those atoms that want more electrons or want to get rid of electrons, and they come together and they share electrons with each other. And so that way both of them are happy. We have non-polar covalent bonding, where the covalent bonds equally share the electrons. This is an example like any of our elements, so 2 things of the same atom put together. And we have polar covalent bonds, meaning that they are sharing the electrons unequally. So one of the atoms is more electronegative, meaning that it has a stronger pull on the electron. So for example, H2O is a big one, where the hydrogens have a weaker pull on the electron, and the oxygen has a very, very high pull on the electron. So the electronegativity is going to be more towards the oxygen in that molecule. Then finally, we also have hydrogen bonding, and that is when hydrogen atoms form polar bonds with the stuff around them. We talk a lot about hydrogen bonds in many of the processes, the different things that we have in the body, from DNA to all of our different protein formation, as well as just essentially just kind of holding us together as adjacent H2O molecules. Again, the human body is made up of anywhere from 60 to 70-something% of H2O. And so you see hydrogen bonding is imperative for keeping us actually together. Okay, so talking about water as a solvent, we have a couple different interactions. We have hydrophobic interactions. Hydrophobic compounds are insoluble in water. Hydrophilic compounds are soluble in water. Hydrophilic, hydrowaterphilic, loving, hydrowaterphobic, fearing. And you can also flip these around and talk about fats as well. So lipophobic and lipophilic. Now, you'll hear Dr. Sarkar all the time talk about like dissolves like. So water is a polar solvent. And so if you put something that has polar bonds in it, it's like dissolves like, and it's going to break it up. Okay. Amphipathic molecules. These are chemical compounds that have both polar and nonpolar regions, typically slightly bigger molecules, and parts of them are polar, parts of them are nonpolar, so they can interact kind of with both sides of that spectrum. So they have both of those hydrophilic and lipophilic properties. Lipophilic is the same thing as saying hydrophobic. Hydrophobic, if it's hydrophobic, it's lipophilic. If it is hydrophilic, it is lipophobic. So those kind of go opposites of each other. Such things as surfactants, the phospholipids, and bile acids. Okay, tonicity. This is talking about a solution. Specifically, a solution is, again, a solute and a solvent put together. So a solute mixes into a solvent. And depending on what the concentrations of the stuff is inside versus outside, is what we'll see movement. So specifically, if we put a cell inside with a hypertonic solution, there's less concentration inside the cell, and so the water goes out of the cell. So the cell is going to shrink and dry up. Let's look at that. Cell shrinks. Hypotonic, remember hippo. It's going to expand because there is there's more concentration of stuff inside the cell than outside the cell. So the water is going to go in to try to balance out the concentrations. And then isotonic means that it is balanced. Iso meaning the same, tonic being the tone. So they're equal inside and out cells, so there's not going to be any real flow or exchange of water. Okay, now getting into acids. So acids, these are the compounds that release protons into a solution. They are considered proton donors. There is another definition for this, but this is the definition there's another definition for acid. But for the purpose of this class and for biochemistry generally, this is the definition that we're going to use. That the compounds that release a hydrogen atom in a solution, releasing a proton, they are proton donors. So weak acids do not dissociate or do not completely dissolve in solution. So it doesn't give all of its hydrogen ions away. Some acids like carbonic or phosphoric acid are examples of that. Strong acids, these dissociate completely in solution. So if you put these in water, all of the hydrogens get released. So hydrochloric acid and sulfuric acid. If I do recall correctly, hydrochloric acid is actually what is often put into pools. That's why when you put in fresh chlorine into pools, you have to wait a certain amount of time before you're allowed to swim in it because it dramatically changes the pH, and there needs to be time for that whole thing, all the water to circulate and to mix, to allow the different compounds to bind and to neutralize, which is why you have to wait a certain amount of time after putting in chemicals in pools. Okay, now talking about bases. These are compounds that either accept protons or they lower the hydrogen or the proton levels of a solution, or they could release hydroxyl ions or OH. These OHs, if you release an OH, the OH is going to find a hydrogen, it's going to bind it, it's going to form water, so then it neutralizes it. Same idea. Weak do not dissociate completely, and strong does dissociate completely. Often you'll see some sort of metal, like a positively charged metal hydroxide, as a strong base. Okay, when we talk about buffers are composed of a weak acid and its conjugate base. So the weak acid is the proton donor that doesn't completely dissociate, and conjugate base equals what is left. It's kind of you say it's the molecule that is left behind when a hydrogen or a proton comes off of the weak acid. So for example, if A is my acid and it has a hydrogen bound to it, we add it into some water and it's going to separate. So now we have just the A by itself and the hydrogen has come off and we still have H2O, we have water in there. This now, without the hydrogen, is our conjugate base. So kind of the way if we talk about neutralizing acids, we'll talk about kind of titration and that kind of stuff. Neutralizing acids, you typically will add more of the conjugate base by itself, because with more of the conjugate base, because this is actually this reaction can go both ways. So if you add a bunch more conjugate base, it's going to push the reaction back this way, forming it and again having it all be neutral and decreasing the pH. We have a couple of different buffer systems. The bicarbonate buffer first. Let me pull up a good image for us. We don't spend a ton of time talking about these in class, so I'm not going to spend a ton of time here, but you will see quite a bit more of the bicarbonate buffer and other buffers in future classes, like Phys 2 focuses a decent amount on the bicarbonate buffer. So the bicarbonate buffer, its function is getting CO2 out of the tissue into the blood, kind of switching it with getting the CO2 out of the tissue into the blood so that way it can go into the lungs and we can actually breathe it out and regulate our pH in the body. So we can see here how within side of the tissues we have breakdown of all this stuff. We get formation of CO2. CO2 comes over here. It gets broken down into HCO3 bicarbonate and is transported in the bloodstream as bicarbonate. That way it's not terribly messing with the pH and we still have it, we have a way that we can transport it without messing too much with the pH of the body until it gets to the lung, and then it'll break down again back into CO2 and hydrogen, and the CO2 can continue and be breathed out. Not a whole lot to be said there. Hemoglobin buffer. Again, this is another one that we don't really talk about this a whole lot. Let me see if it'll. But again, it kind of forms has a similar role. So as a buffer, the hemoglobin counteracts any rise in blood pH by releasing hydrogen ions from a number of atomic sites throughout the molecule. So again, it's just for balancing pH. pH is extremely important in the human body. You'll talk more about this in Phys, but the human body can only really survive with a pH between 7.35 and 7.45. So if we get anywhere out of that range, a lot of our enzymes and proteins start to denature, start to shut down. And it's all kinds of issues. So that's why pH is and focusing on pH and the hydrogen and everything in that concentration, those concentrations are so important for the human body. Finally, the phosphate buffer system. Again, the phosphate buffer system is another type of chemical buffer system in the to regulate the pH of the blood. It's all about the blood. All about the blood. Okay, we talked strength of acids and bases. We already talked about that. Define and know pK values. So pK is the number representation that is used to compare the strengths of different acids. So this is something that in a lab they calculate this. So they use all their fancy stuff to put different acids into different solvents, see them dissociate, how much of them dissociate, and they measure exactly like what the reaction speed and how much of the reaction occurs in order to measure them and to quantify them as, are they weak acids or are they strong acids? That's what the pK value is representing. Not super important to just remember these numbers, but you can if you want to. It really won't come up very often ever in any classes. Knowing the pH scale, so pH, understanding high pH is basic, low pH is acidic. If you have a really high pH, you have very low or no amount of hydrogen. If you have a low pH, you have lots and lots and lots of hydrogen. And there's that normal pH for the blood again. The Henderson Hasselbach equation, just on knowing this is the equation itself, you'll be able to recognize the equation. It's like the I think if I don't remember quite what pH is. I think it's like the partial pressure of the hydrogen or something like that. Equals pKa plus the log, and that is the A minus it is the conjugate base. So the concentration of the conjugate base over the concentration of the acid. That equation relates pH, pKa, and concentrations of the different buffer systems. And at equal concentrations, the pH equals the pKa. I don't know what I'm thinking. Partial pressure. It's pH is pH. My brain was not computing that for a sec. Okay. So know the relationship between pKa values and pH with the concentration of the reactants and product formed. Again, we just talked about that, where when the conjugate base and the acid, weak acid, are identical, the pH is equal to the pKa. Okay. Define and know oxidation and reduction processes with simple examples. We've talked about oxidation and reduction reactions for literally everything. Okay? So hopefully this is very, very kind of ingrained in you. Oxidation and reduction. We have the mnemonic oil rig. Oil rig. Oxidation is loss of electrons. Reduction is gain of electrons. That's in general. You can say it's the loss of electrons, loss of hydrogen, or the gain of oxygen for oxidation. It happens a lot when you have covalent bonds. The formation of H2O. So you think about the oxygen has stolen electrons from the hydrogen atoms, so the hydrogen has been oxidized by the oxygen atom. We have something that's called, with oxidation, we have what's called reducing agents. So the reducing agents are where it's the reducing agent is what it gives the electrons to. So for example, when we talk all about the Krebs cycle, the formation of NADH, right? So NADH, we have NAD plus, and then from the glucose, or once the glucose and everything is split, we're going to take a hydrogen away from that and make ADH, and it's an oxidation reaction. So we oxidize in order to produce NADH. So now reduction is the gain of electrons. In the case of, for example, the water, the oxygen is reduced by the hydrogen because it receives electrons. So we gaining of electrons, gaining hydrogen, or loss of oxygen. Again, you kind of see this oxidation, gain of oxygen, reducing loss of oxygen. Okay. The oxidizing agents accept electrons and allows for reduction to happen. Okay, this next part, to know and identify oxidized and reduced forms of the molecules. These right here, you'll always have a couple of questions talking about these. Just understand these steps. You have primary and secondary alcohols. The primary alcohols get oxidized to aldehydes. The secondary alcohols get oxidized to ketones. This is just straight memorization. Aldehydes get oxidized to carboxylic acids, and carboxylic acids are reduced. So we get oxidized one way, we get reduced going the other way, reduced to aldehydes. And aldehydes then are again reduced to primary alcohols, etc., etc., etc. Basically, these just can go back and forth. So primary alcohols to aldehydes, aldehydes to carboxylic acids. Secondary alcohols go to ketones. Other important chemical reactions. These won't pop up a ton. These will be more important in biochem too. Don't worry about these a lot right now. All of the functional groups. Please, please, please review the functional groups. You don't need to necessarily know the chemical formula of every single one of these, but understand like the big ones. Like you'll want to remember hydroxyl. Like what is a hydroxyl? The hydroxyl is typically the OH. Disulfide can be important. The amino or the amine, you should remember that one as well. The methyl, because methylation is super, super prominent. Yeah, those are the main ones to remember. You might want to remember phosphoryl as well. Okay, that was all of lecture 1. On to lecture 2, the amino acids. Again, hopefully this has been beaten to death with you guys, so it's not too much to review. So knowing the structure of the amino acids, we have the central alpha carbon, we have the carboxyl group, the amino group, and then our R group, which really determines the properties of our amino acid. So talking about carbons, what is a chiral carbon center? It is a carbon that is bound to 4 different groups. This is when we'll pop this question in right here. Which of the amino acids does not have a chiral carbon center? And that is the glycine, because it is the smallest and it has 2 hydrogens. Because its R group is a hydrogen, so it's just hydrogen, hydrogen. So it doesn't have a chiral center. Know what is zwitterionic structure of amino acids, the ionized structure of the amino acid. So look for the one where it has multiple charged groups on it. So it's going to have both a positive and a negative charge, so it can act with both. So if it can act zwitterionic, it can interact with other positive charges, or it can interact with negative charges. It has both of those properties. It also can act as both an acid and a base. Okay, meaning that it can be a proton donor or a proton acceptor. It can do either. Right there, amphoteric. Just what I just explained. Amphoteric is both acidic and basic characteristics. Now, what is a peptide bond? How are peptide bonds formed? So a peptide bond is a covalent bond, but that happens between amino acids. It just gets a special name because it forms a polypeptide chain. They are formed through polymerization by condensation to form peptide bonds, and also remember that condensation is called dehydration. The amino acids, by their polarity, all of them thrown in here. By their chemical group, some of these have different things that you can include. So the sulfur-containing amino acids, we don't usually talk much about taurine or homocysteine. Homocysteine is kind of thrown in there as well. These are the main 2 that you'll want to know, the cysteine and methionine. Branch chain amino acids. I live on a branch, just remember those. The acidic, like acidity of as and glutes. Our basic amino acids, lysine, arginine, histidine, amide, hydroxyl, or aromatic rings, etc., etc., etc. Yeah, a lot of this, coming up with little mnemonics to help remember these is kind of the best way to do these. I would highly, 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 highly recommend, if you have put off learning a lot of these this trimester, set aside some time over the break to go through these and really learn them. Okay, because they will be huge in Biochem 2, and it is a pain in the butt to try and relearn all of these during going through all that other material. It's expected that you know these going in, as well as in clinical nutrition, a couple of other classes in the future, and especially for boards. Please, please, please remember these, and go and start and review and go through these, because these will continue to pop up over and over and over again, and you'll be much better off if you're able to put them and stick them into your brain now. Okay, especially next try, Biochem 2. Trimester 2 is a very, very, very fast-paced try. There's a ton of information and very difficult classes. Not very difficult, just a lot of information all at once. It's like drinking from it's like putting your mouth right on the fire hydrant, basically, next try. And I want you guys to be prepared for that, okay? And I want you guys to be set up for success. So, take some time over the break, come back through, review all of these, make sure that you have these down, because it will come in big handy next try during Biochem 2. Especially because you'll be more on campus. I believe Biochem 2 is still online, but you guys will also be, once you're here in person, you will be also under the Proctorio browser. That is a lockdown browser. So if you have gotten through Biochem 1 through any means besides hard work, you will be in for a very, very, very rude awakening. So take the time to learn these. Okay, I'll get off my soapbox. Okay, so modification of amino acids by glycosylation. On the O side, so the hydrogen in the OH group is replaced by a carbohydrate moiety, like serine, threonine, tyrosine, and linked to the hydrogen in the NH2 group is replaced by a carbohydrate moiety, such as with asparagine. So this is going through the different ways that amino acids can be modified. This is excuse me. This isn't going to be heavily tested. And to be completely honest, if like I had to choose, I would go I would go through this quickly. Just know that you know all like the vocabulary words, like what the words mean, and move on. Because honestly, the types of questions that will come from here won't be super complex, and there probably won't be a lot of them either. Okay, you can have addition of lipids, phosphorylation, acetylation, carboxylation, oxidation, hydroxylation, and addition of metal ions. So we've talked quite a bit about carboxylation, acetylation. We just talked about with DNA. Quite a bit about DNA, and there's also another one that's not on here. Methylation is another one that happens a lot. Okay, lecture 3. Flying. This is the structure-function relationship in protein. Chapter 18, we just talked about proteins, and I'm all about making proteins. So this is kind of all coming full circle now. So peptide bonds, polypeptide chains, and proteins. So the peptide bond is that chemical bond formed between the two molecules when the carboxyl group of one molecule reacts with the amino group of the other molecule, and it releases a molecule of water. What is that reaction called? Condensation. Also called dehydration. Okay, and the chemical bonds of molecules that all come together in a chain are our polypeptide chains. So when we stack a whole bunch of amino acids together, and we are forming those polypeptide chains as we start to build our protein, we get a polypeptide chain. Finally, the polypeptide chains, or chains, will come together to form proteins. They fold all very, very nicely into all of their different shapes and stuff, their primary, secondary, tertiary, and possibly quaternary structure, in order to form functional proteins. Okay, so we have a couple different structural classification of proteins. We can have fibrous proteins. Those are insoluble and they support structure, like collagen and keratin. Collagen is in a lot of our connective tissues, and keratin is in our nails and skin and hair. Globular proteins. These are water soluble, such as myoglobin and hemoglobin. Just remember the globular are the globins. The integral membrane proteins. These are the proteins that are embedded in the membrane. They both support structure and have a function. They can do one or the other, or do both. They also membrane. So these are our membrane-bound enzymes, receptors, and ion channels. Okay, so primary, secondary, tertiary, and quaternary structure of proteins. I think I go into this. Yeah, it's down here a little bit. But remember, just for a quick review, our primary is our polypeptide chain. Secondary, they start to fold. We have our alpha helixes and our beta pleated sheets. Remember, the alpha helix, we can have a right or a left. Most commonly the right, because it is more stable. Beta pleated sheets, we can have parallel, anti-parallel, or mixed. Tertiary, then we get kind of the combination. So it's like the alpha beta alpha or beta alpha beta, where we get the combination now of all the different secondary components to form, potentially at this point it can form a fully functioning protein, or it can go and combine quaternary. This is when a whole bunch of them come together. So multiple tertiary structures come together to form one larger protein. So the binding in peptides, again, this is kind of repetitive. The secondary structure, the alpha helical structures, the right-handed. The beta pleated sheets, the anti-parallel, are more stable. You also have the parallel, which are less stable, and the mixed, which are decent. Anti-parallel you'll typically see a little bit more of because they're more stable. Proline and glycine in the beta bend and making beta pleated sheets. So when you see a proline and a glycine together within a couple amino acids of each other, because both of these are very, very small, they're very flexible. So that's where we're going to see typically the turns happen in the beta pleated sheets. Okay. Bonding and secondary structures of proteins. Trying to remember the bonds that are happening, or what the major bond is. In most of these, you're going to see hydrogen bonds. So secondary structures, the hydrogen bonds determine it. When we talk about a lot of different structures, the way that they fold and their shape is typically determined by how everything binds together. So the charges of each individual thing inside of it and how those charges are going to interact. Some are positive, some are negative, some are neutral, some have some are polar, some are non-polar. So they're all going to interact with each other and form these different bonds, and that's how they're going to get their shape. Okay. Again, bonding and tertiary structure of proteins. Jumping back real quick. Primary structure, it's peptide bonds. Secondary, it's mostly hydrogen bonds. Tertiary, again, it's mostly hydrogen bonds. Quaternary can be a combination of stuff. It is, again, hydrogen bonds, but it's also and also this is in tertiary structure as well. You can have disulfide linkages. This is when you have those cysteine molecules, those cysteine amino acids, where they'll link together kind of across the aisle from each other and form these disulfide linkages because they are sulfur containing. Denaturation and renaturation of proteins. We talked about this just a minute ago in chapter 18. We also talked about it way at the beginning. The denaturation is the unfolding of the native protein, and renaturation will usually happen naturally if left alone. Denaturation can happen because of extreme heat, changes in pH, as well as stuff like detergents, urea, alcohol, strong acids, or strong bases. If they're in the presence of these sort of things, it'll break them down. And if you move them back into a neutral environment, then they will typically renature. If they get destroyed beyond a point of just like your standard denaturation, then they won't renature. But it takes a decent amount for that to happen. The oxygen binding properties of myoglobin and hemoglobin. Remember, myoglobin is in the muscle. Myo means muscle, and hemoglobin is in the blood. Heme is blood. So you have cooperative binding and significance of the sigmoidal curve produced by hemoglobin during its saturation by oxygen, and compare this with the oxygen binding of myoglobin. I don't have the chart here. Let me find it. Myoglobin versus hemoglobin chart. The sigmoidal curve. So we see here how hemoglobin is a little bit more constant. Myoglobin fills up very quick, gets saturated very quickly. Hemoglobin is more of a gradual process. And this is due to the fact that myoglobin only has one binding site for oxygen, whereas hemoglobin has 4. And that gets into cooperative binding. So the cooperative binding, cooperative binding is that each time an oxygen binds to hemoglobin, it increases the affinity for oxygen, meaning that it wants to bind another. And it also, because if you think about remembering that hemoglobin is actually a quaternary structure, and that it has 4 different proteins kind of all put together. And each side of that the protein as a whole, when it binds an oxygen, it will change its shape. And changing the shape will reveal another binding site for oxygen. And that's what that cooperative binding is. And it's positive cooperative binding for hemoglobin. You can also have negative cooperative binding. So if you have binding of one or more molecules of a ligand, it can make it more difficult for subsequent ligand molecules to bind. Remember the subunits of adult hemoglobin. There's alpha and beta. I think those are the only 2. Let me go check. Yeah. We have the alpha and the beta parts of the hemoglobin. There's 2 alpha and there's 2 beta subunits. Slightly different, a fetal will have gamma subunits because those have a greater affinity for oxygen, because babies need a lot more oxygen because their cells are in overdrive, reproducing and dividing to let the baby grow. So sickle cell anemia, you actually don't get binding of all 4 oxygen because there's a change in the subunit structure. And yeah, actually we just mentioned this a little bit ago about the mutations. In the chapter 18 video, we talked about, I believe it was chapter 18. It might have been chapter 17. But in one of those videos we talked about this, how we have a change of glutamate changes to valine at position 6 of the hemoglobin beta chain. So literally this one single change due to a mutation literally causes all of these issues with hemoglobin. So really important stuff to know, that make sure that our DNA is working properly. Okay, we've got enzymes left. I'm going to do a pause and a quick cut here. I want to keep these videos at right around 40 minutes. That way they're a little bit easier to digest. So I will take a break here, and we will start again in the next video.